We are preparing for the SeekSpace single-cell spatial transcriptome seq experiment. Now we proceed directly to Step 2, preliminary reverse transcription reaction. Prepare all newly required reagents and consumables according to the checklist. Previously prepared wash buffer and 1x PBS-RI from Step 1 are used directly in this step. Before use, make sure all reagents are properly mixed. Reagents, except enzymes, can be vortexed and briefly spun down. Enzymes should be mixed gently by pipetting, then briefly spin down. Prepare the mix on ice. Pipette 15 times to mix. Spin briefly. And keep the mix on ice for later use. The prepared reaction mix is then distributed into 8-strip PCR tubes for multiplex labeling. For each sample, the pre-RT reaction is evenly split into 8 parallel reactions. 17.7 microliters of pre-RT mix is dispensed into each tube. Then add 1.5 microliters of a unique multiplex barcode, ranging from SRB09 to SRB16, to each tube. This multiplexing strategy is designed to increase per-sample input capacity and improve cell nucleus coverage from each spatially labeled tissue section. Prepare the nuclei input for each reaction according to the defined limits. The total nuclei per sample should not exceed 240,000, with 600 to 30,000 nuclei per reaction. The nuclei suspension is first prepared and distributed across the 8-strip PCR tubes. Each tube is then loaded sequentially with the calculated nuclei volume. Each reaction is adjusted to a final volume of 40 microliters, using 1x PBS-RI where required. The same procedure is applied to the second sample, with reactions prepared in parallel. After all additions are completed, the reactions are gently mixed using a multi-channel pipette to ensure uniform distribution. After mixing, tightly seal both 8-strip PCR tubes. Keep the reactions on a pre-chilled metal block and immediately transfer to the thermal cycler. Run the pre-RT program with the heated lid set to 85 degrees Celsius. After completion of the pre-RT reaction, place the tubes back on ice. The 8-strip PCR tubes are briefly opened and each reaction is ready for post-RT processing. Add 1.5 microliters of buffer S to each reaction sequentially. During this process, the reactions remain on ice to maintain stability while all wells are processed one by one. The same procedure is applied to the second sample in parallel. The 8 reactions are then pooled into a single tube for each sample. Transfer each reaction tube sequentially, ensuring complete recovery of reaction products. Before transfer, each tube is briefly mixed to resuspend residual material. Combine all collected fractions into the same pooled sample. Then sequentially rinse each original tube with a total of 100 microliters of wash buffer. The same wash is transferred from tube to recover residual material at each step. Add 1 milliliter of wash buffer to the pooled sample and mix thoroughly. make sure that the wash buffer volume is at least 2 times higher than the pooled pre-RT reaction. The same procedure is applied to the second sample in parallel. Then centrifuge at 1000 G for 5 minutes at 4 degrees Celsius. After centrifugation, carefully remove the supernatant, leaving approximately 50 microliters to keep the nuclei pellet intact. This process is then applied to the second tube, where we again gently remove the supernatant to preserve the pellet structure. The same wash and spin cycle is then repeated twice to ensure complete removal of residual reaction components. Gently resuspend the nuclei pellet in 1 times PBSRI. Adjust the final volume to 90 microliters to ensure sufficient input for downstream quality control and processing. Briefly process the nuclei using the SeekSpace single-cell suspension preparation system. Insert the probe 1 to 2 millimeters below the liquid surface and operate for 1 to 2 seconds per sample. Process each sample individually and clean the probe between samples to prevent cross-contamination. Use a fluorescence counter to determine the nuclei concentration. Adjust the nuclei suspension if needed to reach the recommended concentration range of 2,000 to 3,000 nuclei per microliter before downstream processing. Preliminary reverse transcription is now complete. We will now proceed to droplet barcoding,